Hello, biology superstars! Welcome to the first video for A2.1, all about nucleic acids. In an attempt to break up this content a little bit better, I am going to go over just the theme, level of organization, guiding questions, and associated skill in this video, sort of like an intro. And then I'll do SL notes in another video and HL notes in another video. So if what you're looking for is strictly note content, probably click out of this and keep going. But as I said before, I do think that these associated skills can be super useful, and it's nice to know why the IB has chosen to organize their syllabus into these themes and levels of organization, especially if you're like studying for your actual IB exams. So let's get started. All right, these are all the things that we'll do in this video. First up, theme and level of organization. So we are still in the unity and diversity theme. Biology is the study of life. Life is very diverse, but all life shares many important unifying characteristics. And as we look at nucleic acids, we're actually going to get into the idea of these unifying characteristics and molecularly where they come from a little bit more, because we are at the molecular level of organization. And of all the biomolecules associated with life, nucleic acids, which are the focus of this little subsection of the syllabus, are perhaps the most important. They allow for heredity and the creation of proteins, and both of these processes are essential for both life and evolution. So in my opinion, that's why the IB put the study of these nucleic acids like RNA and DNA in the unity and diversity theme, and the molecular level of organization. Some guiding and linking questions. The guiding questions from the IB are: how does the structure of nucleic acids allow hereditary information to be stored? And how does the structure of DNA facilitate accurate replication? So you'll notice probably both of these questions ask you about structure of molecules. The structure of these nucleic acid molecules is super important, and with like the history of the discovery of the structure is super interesting, and we'll get into that a little bit throughout this content. And I do think in terms of guiding your thinking about nucleic acids as molecules, it is really important and really like fascinating and beautiful to consider their structure, because it allows for so much that is important for life to exist. This DNA needs to be able to replicate itself. It needs to be able to store and pass on information so that life can continue to evolve. And that's, I think, why they chose these as guiding questions. Linking questions, we will learn in this content about the structure of both RNA and DNA. So one linking question is, what makes RNA likely to have been the first genetic material rather than DNA? To be fair, A2.1 is only advanced higher level content, but it does talk about the origin of cells and RNA as the presumed first genetic material. And then the second linking question, how can polymerization, which we'll talk about how DNA and RNA form polymers, the bonds that allow them to create big, long molecules. We'll talk about that in this content. How does that result in emergent properties? And C, 3.1, also talks a lot about the body systems coming together to form emergent properties of life. C 3.1 is actually called Integration of Body Systems. It is a beast. I think there's like 25 points of information, and it deals with the brain and the nervous system and the endocrine system and the respiratory system and the digestive system. So this idea of like small pieces, small systems coming together to form a larger whole is super important in that content also. And thinking specifically about nucleic acids, looking at associated skills, I want to bring up this idea of interpreting data to draw conclusions, because it seems like this is something the IB is placing more and more importance on all the time. Being able to interpret data that is given to you and draw conclusions from it is super important in both the paper 1B and the paper 2A. There are now 4 database questions in paper 1B, and there's like a big long database question in paper 2A. So the data that I thought that we could look at to draw conclusions is the data from an experiment from a scientist called Erwin Chargaff. This experiment is explicitly mentioned in the HL content for this section, but I sort of think that they could like throw this data at you and expect you to be able to interpret it a little bit, even if you're just taking an SL exam. So let's look at a little set of data that could have been drawn from this Chargaff experiment. Erwin Chargaff worked in the 1940s and 50s, primarily at Columbia University. He also studied undergraduate at Yale and did some, I think, post-grad work at Pasteur Institute in Paris and University of Berlin. But he got really, really famous for the work that he did, published in the 1950s. Oh no, there goes my thing. Hold on, I'm going to delete and start again. So 1950s, let's see if this gets a little more agreeable. 1950s at Columbia. And what Erwin Chargaff figured out was the ratio at which different DNA nitrogenous bases are present in DNA samples. So in this data set, you have the 4 DNA bases, adenine, cytosine, guanine, and thymine, all listed out. And then it gives you a bunch of different organisms, and down here at the bottom, a virus. And it gives you the percentage of each DNA base that is present in each organism. So if you take a minute to look at this data, you will hopefully notice that, let's get my pen going, in the majority of these samples, adenine and thymine are present at roughly the same ratio, and cytosine and guanine are present at roughly the same ratio. So what that allows you to deduce, or like the conclusion that you could draw, is that in some way adenine and thymine are paired up together, like they're important to each other, and cytosine and guanine are paired up together, important to each other. This is present in all of the samples. And you'll notice in some of them, the numbers are like a little bit off from each other. So here, this one's 22, this one's 24, here, this one's 19, this one's 18, but they have roughly the same proportions. So first, let's leave viruses out of it and talk about this wide variety of living organisms where this statement is true. I think the IB, for example, could ask, what is the relationship? Like, what is the ratio here? State the trend. And the trend is that A and T and C and G are equal in present, are present in equal ratios. Also, I think the IB could ask, like, what's up with these, where they're a little bit different? And I would say the places where they're a little bit different, the differences, once you've studied DNA a little bit, differences could be accounted for by the idea that there are mutations and occasional mistakes in DNA replication. Mutations, mistakes. And then obviously, probably you could ask, like, what's up with the virus? Why doesn't this follow the same ratio? And obviously, when you're looking at that, you would say maybe this gives evidence that viruses are not living. Viruses do not equal living. You could also ask why all of these percentages don't seem to add up to 100. And as I'm looking at this, I'm realizing that I think I just made some like simple math mistakes. But let's just pretend they all add up to 100. And I think that that is a good way to interpret data and be able to draw conclusions from it. So in a paper 1B, the IB would ask you this, like sort of knowing that you have some experience with looking at like what these, like what molecule is represented here. You know that it's DNA. What like draw a nucleotide of DNA because they give you stuff that they know that you've studied a little bit about, and they'll also ask you content-based questions. In a paper 2A, that like which is where it maybe would be more likely to come up on an SL exam because you're not like explicitly taught this in the SL curriculum. That's where you could be given this data as like an unfamiliar data set and just asked to interpret it like what's going on here. All right, so I hope that this was helpful in terms of thinking about database questions on IB exams, and I will see you for the SL content in the next video.